Hello everyone, welcome to my channel Anupma Biology Classes. This is the first video in the series of reproduction or embryology. Today our topic is reproduction in organisms in plants and animal both, and this is the introductory portion of the embryology. After this, from the next video, we will understand the details of all the process and organs of the reproduction in plants and animals. Before this video, we discussed on the human physiology and cell biology. So this is the third series of my biology classes, and this video is from CBSE class 12th chapter 1st. So let's start the chapter which is related to the reproduction. So the question is, what is the reproduction? Reproduction is one of the most important process of the living organism to make a copy like itself. And reproduction is the biological process by which new individuals, organisms, that is the offsprings, are produced from their parents. It is important for the survival of all the living things, and each individual organism exists as the result of reproduction. Now, reproduction is of two types, in which the first one is asexual reproduction and the second is sexual reproduction. So in case of asexual, there is no fusion of gametes and new individuals are produced by single parent. While in the sexual reproduction, there is fusion of gametes and the new individuals are produced by participation of two separate parents. Now, you can see the main changes in sexual and asexual reproduction. So in case of the asexual reproduction, it involves only one parent, while in case of the sexual reproduction, it involves two parents, that is male and female. In asexual reproduction, gametes are not produced, while in case of sexual reproduction, gametes are produced by the organism. In case of asexual reproduction, offsprings are genetically identical to the parent, while in the sexual reproduction, offspring show genetic variations. In asexual reproduction, the cell division are only mitotic, while in the sexual reproduction, gametes are produced by the meiotic divisions and the zygote developed by mitotic division. In asexual reproduction, a large number of plants or animals are produced in a very short time, while in the sexual reproduction, comparatively, the number produced is less. Now, the first part of the reproduction, that is the asexual reproduction. And the asexual reproduction occurs mostly in the unicellular organism like monerans and protists, and in plants and certain animals also. Here, young ones receive its genes from one parent, so it is known as agametesis or agamogeny. And here it is a kind of reproduction which involving only one parent, in which meiosis, gamete production, fertilization, transfer of genetic material between the two individuals, and parthenogenesis doesn't occur. It produces identical offsprings called clone. It involves only mitotic cell divisions, which termed as somatogenic reproduction. Now, the types of asexual reproduction. And there are different ways for the asexual reproduction, in which the first one is binary fission, where the division of parents into small two parts nearly equal sized daughter individuals. And the examples are, first one, that is the binary fission in bacteria, binary fission in protozoan protist, and the binary fission in case of planarians, which we will discuss here in detail. So this is the first picture here, the binary fission in case of bacteria, where the cytoplasm and nucleus divides into two without mitosis. And during the division of nucleate, the DNA replicates while attached to mesosomes. And in this phase, the cell wall and plasma membrane start growing transversely inwards from near the middle of the dividing cell between the two mesosomes. Now this divides centripetally and separates the parent cell into two nearly equal daughter cells, each having a nuclear body. This process is under favorable condition, completes in about 20 to 30 minutes. So in 24 hours, large number of bacterial cell produces. Next is in amoeba, binary fission is irregular type. Here the plane of division of cytoplasm is variable but is always at right angle to the elongated dividing nucleus. Next, in Euglena, the longitudinal binary fission occur, and here the cytoplasm is splits in length, forming two similar daughter individuals. Now, in ciliates like Paramecium, transverse binary fission occur, and here the cytoplasm divides transversely between the two sets of nuclei, forming two dissimilar daughter individuals. Now, this is the picture of planaria. Here you can see the regeneration. In planaria, both occur, that is binary fission and regeneration. In planaria, divides by transverse binary fission. In planaria, regeneration also occurs because in planaria, you can see a cut into pieces and each piece can regenerate into a complete organism like the picture. So, what is the regeneration? Regeneration means that an organism regrow a lost part so that the original function is restored. Now, the second asexual reproduction, that is the multiple fission. Multiple fission is the division of parent into many small daughter individuals simultaneously. And here are the examples. The first one in case of the Plasmodium. In the picture you can see Plasmodium is a malarial parasite which shows the multiple fission. Here the active adult also known as zygont as well as in encysted zygote known as sporont. And the process is known as schizogony in active adult and in zygote sporogony. And here after the formation of daughter cells it known as merozoites and sporozoites respectively. Second is multiple fission in case of amoeba. In amoeba, the products of multiple fission become individually surrounded by the resistant coat, and this coat is known as cyst wall before the release from the parent. Here, the multiple fission is called sporulation and the encysted products are spores. In favorable condition, each cyst hatch into a small amoeba which gradually grows into an adult. Now, the third asexual reproduction, that is the budding. Budding is the formation of a small individual from a small projection, that is the bud, which arises on the parent body. So the first example is budding in case of yeast, which you can see in the picture. Yeast is a unicellular fungi and it reproduces by the formation of spores. In this process, the parent cell develops a small protuberance which grows in size and the parent nucleus divides and one of the two daughter nuclei goes into the bud. Finally, the bud gets detached and grows into a new individual. So this is the process of the yeast. Second example in case of the hydra. In hydra, buds arises from the surface of the parent's body and each bud grows and gradually assumes the form and size of the parent. When full grown, the new individual may separate from the parent and become an independent animal. So in case of hydra, you can see after the growth of bud develops mouth and tentacles. Then the bud detached from the parent and becomes independent. Now, the fourth is that is the spore formation. Spores are the propagules which germinate to produce new individual. These are the following types of spores in which the first one is zoospores. The reproduction by zoospores occur in some lower fungi, that is the Phycomycetes. Second is sporangiospores and the examples are Rhizopus and Mucor. In this figure you can see the sporangium which forms the sporangiospores. Now the chlamydospores and the example is Chlamydomonas monas. Next is Oidia and the example is fungus, and after that the Conidia example is Penicillium. Here you can see the conidiophores which is the aerial hypha which forms the conidiospores. Now we see the detail of all the pictures here. So these are the various type of spores in which the first one is the zoospore, which are motile and flagellated spores produced inside zoosporangia. And here the flagella help them to swim in aquatic habitat for the proper dispersal. Next is conidia. which are non-motile spores, produced singly or in chain of constriction at the tip or lateral side of special hyphal branches called conidiophores, and they are produced exogenously, dispersed by wind, and germinate directly by giving out germ tubes. Next is chlamydospores, which are thick wall resting spores produced directly from the hyphae cells, and they store reserve food material and are capable of withstanding and giving offsprings in the unfavorable conditions. Next is oidea, where the hyphae breaks up into its component cells or the small pieces which behave like spores, and these are usually formed under excess water, sugar, and certain salts. They germinate immediately after liberation and cannot survive under unfavorable conditions. And the last is sporangiospores, which are non-motile spores produced in individual in the sporangia, and they are dispersed by wind and germinate to produce new mycelium. So these are the complete structures of the spores. Now the next is gemule formation. Gemmules are the dormant structures produced asexually by the formation of specialized exogenous buds at certain times of the year in many freshwater sponges and a few marine species. This is the structure of the gemmules, and here the archaeocyte accumulates nutrients by the phagocytizing other cells and then clustered together within the sponge. Here, certain cells surrounding each cluster then secrete a thick protective covering which is known as gemmule, and after gemmule formation, the original sponge disintegrate, and in favorable condition, floating gemmule produces a new sponge. Now, the next is vegetative propagation. Vegetative propagation is a form of asexual reproduction occurring in plants in which a new plant grows from a fragment of the parent plant or a specialized reproductive structure. Many plants naturally reproduce this way, but it can also be induced artificially. So, the new plant is genetically identical to the parent, which is of two types, in which the first one is natural vegetative reproduction and the second is artificial vegetative reproduction. Now, the methods of vegetative propagation, in which the first is natural method, and here the first is root in natural method, and the examples are sweet potato, tapioca, yam, dahlia, tinospora, etc. So here these are the modified different roots which you can see in the picture, that is the conical root, fusiform root, napiform root, tuberous root, and nodulated roots. These are the modifications in case of the root. And here in Dahlia and asparagus you can see the modified root, and these roots are tuberous which can be propagated vegetatively when planted in soil. The buds present on the roots grow into leafy shoot above ground and adventitious roots at their bases. So each slip, that is leafy shoot, give rise to a new plants. Here the roots modified for the storage of food. Here the three examples you can see, that is the conical, napiform, and the fusiform. Conical roots modified taproot of the conical shape throughout which they store food, and it is broad at the base and gradually tapers towards its apex like the carrot. Napiform roots are the modified taproots, fleshy with the upper portion inflated or swollen and abrupt narrowing of the base into a tail-like portion. Examples are turnip and beetroot. And next is fusiform root, which is modified taproot with thickened middle portion containing food and tapered towards both ends. And the example is radish. Now, underground stem. These are of following types. Like the suckers, the example in case of the mint and chrysanthemum, you can see in the picture. Next is rhizomes, and the examples are typha, canna, ginger, turmeric, lotus, saccharum, etc. Next is corms, and the examples are crocus, gladiolus, colocasia, etc. After this, bulbs, examples are onion, lilies, garlic, etc. And next is tubers, examples is potato tuber. So please see here the detail of the underground is stem, and in this picture you can see the eye bud, which is a tuber, and each eye bud grows into a new potato plant when planted with a portion of the swollen tuber. Potato crop is raised by the tubers, not by seeds. Second is bulb, which produces new plants when sown in soil. Next is corm, which you can see in the picture, which is a highly condensed and specialized underground stem which bears many buds and produces new plants in favorable condition. Next is rhizome, which means vegetative propagation by perinating under unfavorable condition and producing new aerial shoots during the favorable season. Next is sucker, which arises from the base of erect shoot, grow horizontally in the soil, and then come out to form new aerial shoot. These shoots become independent plants and when suckers break away from the parent plant. Now, creeping stems, and these are of following types. First one is the runners. Examples are cynodon, oxalis, centella. Next is stolons, and the examples are Fregeria, centella. And then the offset, and the example is pistia. In this picture, you can see all the creeping stems structure, and in here the runners in the picture you can see which produces adventitious roots at nodes, and each node give rise to aerial shoot which become a new plant. In case of stolons, which are arched runner, which cross over small obstacles and develop small plantlets at their nodes. Then suckers are the underground stem which we discussed earlier. And next is the offset, which are the internode long runner which develop tuft of leaf at the apex. Now the aerial stem. Aerial modified stem of cacti develops new plants when stem segments fall on ground and act as means of vegetative propagation. Examples are the opuntia. In this picture you can see the aerial modifications in case of the stem. Now leaves. Examples are Bryophyllum, Digrymotium, Begonia, Streptocarpus, Santa Paula, etc. And here in this picture, some plants develop adventitious buds on their leaves which get detached and develop new plants. Like you can see in the bryophyllum, which develops new small plantlets with adventitious roots along their margin. Next is bulbils, and the examples are oxalis, Allium sativum, Dioscorea, lily, pineapple, etc. These are the fleshy buds produced in the axil of the foliage leaves in place of axillary buds, and they grow to form new plants when shed and fall on the ground. Then turions, and the example are aquatic plants like Potamogeton, Utricularia, etc. You can see in the picture these are the special fleshy buds and that develop in aquatic plants for the vegetative propagation. So these are the complete structure of the turions, bulbils, leaves, aerial stems and the creeping stems. Now the artificial method of vegetative propagation, and here these artificial methods are man-made propagation and used for the commercial production of crops. So some of the artificial methods are given below. Cutting. Here the small piece of any plant organ used for the propagation, and the examples are the begonia, glycinia, coilus, duranta, rose, sugarcane, etc. In rose plant, propagation is occurred by cutting. In sugarcane, also propagated by cutting, and these stem cutting are used for the artificial vegetative propagation. About 20 to 30 centimeter long piece of stem are planted in the natural position in the soil for the proper sprouting. Here In case of cutting, the factors like age of parent plant, length and diameter of the cutting, season and the type of plants are taken into consideration for the propagation of a particular species. Here you can see the propagation with root cutting. Root cutting is used in citron and tamarind. For the proper development of roots, sometimes stem cutting are treated with IBA, that is indole 3-butyric acid, that is a plant hormone. Now, the second is that is the layering. In this method, roots are artificially induced on the stem branches before they are detached from the parent plant for the propagation. It is of two types. Here in the picture you can see the simple layering and the mound layering. So in simple layering, which is natural layering, occur when the branch touches the ground whereupon it produces an adventitious roots. While in mound layering is in which the various woody stemmed plants are cut back to the ground in early spring and the new shoots that they develop are covered with soil to a depth of 6 to 8 inches to induce root growth, which forms individual plants that can be removed in the fall. So in two types, first one is the mound layering, which is most common method of propagation in the herbaceous plants like the jasmine, grapevine, strawberry, raspberry, etc. And the second is air layering or guti. This method is completely employed in case of shrubs and trees which do not possess branches near the ground. And the examples are lychee, pomegranate, orange, lemon, locust, etc. So in this picture, first is tip layering, which is natural and tip can be used for the propagation. Second is mound layering, in which the lower branch of the stem is bent down and partially defoliated. An injury is made in defoliated portion and this portion is covered by moist soil that growing tip of the branch remains above the soil surface. Within few days, the plugged portion develops adventitious roots. And the last is air layering, where the ring of bark is removed as a slit act as an upward single is made of the base of an aerial branch. Here, the girdle portion is then covered with moist moss or grafting clay and wrapped with a polythene sheet. This wrapped portion is called guti. In sheet the picture you can see the circular portion of the air layering which shows the guti. Now in this picture first is simple layering while in the mound layering you see the bent down and partially defoliated portions, and then in the air layering you can see the first bark tissue removed, then the moist soil placed on that portion, then the polythene tied to retain moisture, and here after this, you can see the new plants by the propagation technique, which is the artificial method of vegetative reproduction in case of plants. Now, next is grafting. In this method, parts of two plants are joined in such a way that they grow on same plant. The rooted supporting portion of one plant is stalk, which is jointed with twig of another plant called zygote. It is of four types. First is the tongue or whip grafting. Both the stalk and zygote cut obliquely at about the same angle, which you can see in the picture. This is the tongue grafting. Then the wedge grafting. A V-shaped notch is made on the stalk and a wedge-shaped cut is made on the zygote. This is the wedge grafting. Then the crown grafting. Several zygotes having wedge-shaped cut are grafted on the sides at the top of the stalk, and which you can see in the picture on the stalk there are several zygotes are placed. It is known as crown grafting. And then the side grafting, where the single zygote having wedge-shaped cut is inserted in the lateral slit of the stalk. So the examples of the grafting are the mango, rose, apple, rubber, citrus, pear, plum, peach, etc. This is the side grafting which you can see in the picture. So in tongue and wedge grafting, the zygote and stalk have almost the same diameter, while in crown and side grafting, the stalk have more diameter than the zygote. Next is bud grafting. This method is similar to grafting except that zygote in this case consists of a bud along with a small portion of bark having intact cambium. Example are rose, apple, peach, etc. In this picture, the living tissue of zygote bud is inserted into an incision made in the stalk so that it reaches up to the cambium of stalk, and this bud should exposed outside in the air. This bud will grow after 3 to 5 weeks later. Next is propagation by plant tissue culture. This is also known as micropropagation. This method includes propagation of plants by culturing the cells, tissues and organs known as tissue culture. Here, tissue sample is scraped from the parent plant. Then the tissue sample placed in agar growth medium containing nutrients and auxins. Then the sample develops into tiny plantlets and then the plantlet planted into compost. This is the complete process of the tissue culture. Now, what is the use of tissue culture? Tissue culture is most important for obtaining virus-free plants, disease-free plants, homozygous diploids, and in commercial micropropagation of orchids and carnations, gladiolus, chrysanthemum, and other ornamental plants. So this is employed for the quick multiplication of plants. Now, the importance of vegetative propagation, in which it is the only method of the reproduction in those plants which have lost their capacity to produce seeds. It produces a small quantity of viable seeds, and plants which have poor seed viability or prolonged seed dormancy reproduce by this method. Because this method are more rapid and here the less expensive also. So the last greatest advantage of this method of the plant is biotype can be retained and multiplied indefinitely without any changes or variations. Now, the sexual reproduction. It involves the production of male and female gametes either by the same parent or by the different parents. When the two opposite types of gametes are produced by the same individual, known as bisexual, and when both the gametes produced by the different parents are known as the unisexual. Male and female gametes fuse to form the zygote, and it is a kind of reproduction that occurs between two individuals involving meiosis. So in this picture you can see all the vegetative phases, and here the first you can see like the embryo formation, that is the embryonic phase, then the period of growth between their birth up to their reproductive maturity which is known as the juvenile phase, then the period of growth between the seed germination up to the initiation of flowering is known as vegetative phase, and the last is when the organism start reproducing sexually which is known as the reproductive phase. So these are the vegetative phase changes is the transition from juvenile to the adult growth in case of plants. Now the events in the sexual reproduction, and here there are three events. First is the pre-fertilization events, then is the syngamy or fertilization, and the last is post-fertilization events. In case of pre-fertilization events, there are two processes. First one is the gametogenesis and second is gamete transfer. While in case of post-fertilization events, also two events. First one is the zygote and the second is embryogenesis. Now, the first, that is the pre-fertilization events. This is the preparatory stage which have two parts, in which the first one is gametogenesis. Here the process of formation of two types of gametes, that is male and female, inside the gametangia. In this picture, you can see all the isogamy, anisogamy and oogamy, that is a type of gametogenesis. Here, all the gametangia where the gametes are formed is known as the reproductive unit, that is the gametes. And in the picture, the first one, that is the isogametes or homogametes. Here, the two gametes are morphologically similar in shape and size. Second is anisogamy, where the both male and female are morphologically distinct. And the third is oogamy, where the both male and female are morphologically distinct, but the male is microgamete and motile, while the female is macrogamete and large and non-motile. In human, microgamete is sperm and mesogamete or macrogamete is egg. So this is the type of gametes formation. Now, if male and female gametes are produced by the male and female parent respectively, called unisexual or dioecious. If male and female gametes are produced by a single individual, called bisexual, monoecious or hermaphrodite. And here you can see the cell division during the gamete formation in this picture. And in this picture, their gametes are usually formed by the meiotic division and are haploid. So it means the equational division, which join to form zygote, which is diploid, and it forms after fertilization. Then this zygote are give rise to offsprings by the mitotic division. So offsprings are diploid like parents, which formed haploid gametes by the meiosis for its creation. So this is the complete process. And here now you can see the second, that is the gamete transfer. Once the male and female gametes are formed inside the gametangia, they are allowed to come in contact with each other to facilitate their fusion. In this picture you can see the male is motile, and during this transfer of gamete from male to female, large number of sperm fail to reach the female gamete. So, to compensate this loss of the male gamete during transport, they are produced in several thousand times at the number of female gametes. So, male gametes are produced always in large in number as in comparison to the female. In this picture you can see the pollination, because the male gamete is non-flagellated in case of higher plants, and they are produced as pollen grains which produced in microsporangia and transfer with the help of water, air, animal or birds. In case of animals, male gametes are transferred to the female gametes by the three distinct patterns. The first one is external fertilization and external development. It occurs in case of obelia, nereis, labio, frogs, etc. Here, the parent releases sperms and eggs into the water, where the fertilization occurs and the zygote develop into offspring. Second is internal fertilization and external development. It occurs by the cloacal apposition in birds with modified arms in cuttlefish, etc. Here the sperms are passed from the male into the female with an intromittent organ like the penis, as in shark and lizard. Here a female reproductive tract provides protective place where sperm and ova can easily meet. Then the zygote passing down the female reproductive tract to the exterior can acquire secrete membrane or cell for the protection of the developing embryo. And the last is internal fertilization and internal development. It occurs in case of the mammals. Here the internal development provides additional advantage to embryo. As the mother carries the embryo whenever she goes, it is not vulnerable to predators who attack externally to developing eggs. Here in this picture you can see the fertilization which occur outside the body, that is known as the external fertilization, and the fertilization which occur inside the body, which is known as internal fertilization. So There are many differences between the external and internal fertilization, which you can see here. In external fertilization, male gametes swims to the female gametes, and here also the male gametes swim to the female gametes in internal fertilization. In external fertilization, no copulation, while in the internal fertilization, copulation occurs. In external fertilization, the male gametes are shed into a large space, there is less chance of fertilization. While in the internal fertilization, the male gametes are shed into a confined space, there is a greater chance of fertilization. In external fertilization, many female gametes are produced, while in the internal fertilization, few female gametes are produced. This saves body material. In external fertilization, zygotes develop outside male and female partners, while in the internal fertilization, zygote can be retained inside the female body for the protection until it is fully developed. In case of external fertilization, most common in fishes, amphibian and algae, while internal fertilization occur in the land plants, reptiles, birds and mammals. So these are the complete differences between the external and internal fertilization. Now we come on the second, that is the syngamy. Syngamy and fertilization. Syngamy is the fusion of two entire gametes to form a zygote. With regard to the resource of the fusing gametes, it is of two types. First one is the endogamy, which involves self-fertilization, which is uniparental. Example is tinea. And the second is exogamy, which involves cross-fertilization. It is biparental, and the examples are frog, rabbit and man. Next is, with regard to the structure of the fusing gamete, it is of two kinds. First one is isogamy. The fusing gametes are similar morphologically as in monocystis of protozoan. Here, the gametes are known as isogametes and their fusion is known as isogamy. Next is anisogamy or heterogamy. The fusing gametes are different in form, structure and behavior as in frogs and humans. And here the gametes are known as anisogametes or heterogametes and their fusion is known as anisogamy or heterogamy. In this picture you saw anisogamy in case of humans because you can see here the male gamete, that is the sperm, is smaller in size while the female gamete, that is the egg, is larger in size. Male is motile while the female is non-motile. And in case of the plant, in the ovary the pollen attached with the stigma and enters into the ovary with the help of a style. It shows triple fusion, which we will know in detail in the next video. Now, parthenogenesis. Parthenogenesis is a modification of sexual reproduction in which an egg develops into a complete offspring without fertilization. It is monoparental. It is of two main types. The first one is natural parthenogenesis, which is a regular phenomena in life history of some animals. It is of three types. First one is complete parthenogenesis. It occurs in some rotifers and few vertebrates. There are no males at all. So, female develops exclusively by the new offsprings, like the caution rock lizard. Next is incomplete parthenogenesis. Here the some animals have both sexual and parthenogenetic individuals, which may alternate, and the examples are honeybee, daphnia, etc. So in this, the female can produce fertilized or unfertilized eggs depending upon their environmental condition. And in honeybee case, the unfertilized eggs develop into male bees, that is drone, with haploid cells, which you can see in the picture. Drones are n in number chromosomes, that means haploid condition, while the fertilized eggs give rise to female with diploid cells, that is 2n condition, where the workers are the fertilized sterile female and the queen is the fertilized female, and the workers are 2n number and the queens are also 2n number chromosomes, that means diploid. So drones are haploid and the females are diploid. Next is pedogenetic parthenogenesis. In certain insects, larvae lay eggs which develop parthenogenetically into a new generation of larvae. Second type of parthenogenesis is artificial parthenogenesis. Here, the eggs of certain animals such as annelids, mollusks, starfish, frog, hen, rabbit, etc. can be induced to develop parthenogenetically by artificial stimuli. So, what are the artificial stimuli? Artificial stimuli like the prick of needle, electric shock, changes in the temperature or pH, addition of urea, fatty acid, ethers, chloroform, etc. Now, the post-fertilization events. Here, the events which occur during the sexual reproduction after the fusion of gametes is known as post-fertilization events, in which the first one is the zygote. Here, the fusion of two haploid gametes results in the formation of single diploid zygote. It is the first cell of the diploid organism, including human beings. And the second process is embryogenesis, where the process of development of embryo from the zygote. So, in this flowering plant, you can see the zygote develop into an embryo inside the embryo sac. And here the ovule transform into seed and the ovary become into fruit. Here you can see the fruit develops from the following layers: fruit wall from the ovary wall and seed from the ovule. In the animals, the development of zygote takes place either inside the body of female parent or outside. On this basis, animals are grouped into two categories. First one is oviparous animals. These animals lay fertilized or infertilized eggs like reptiles and birds. And the second is viviparous animals. These animals give birth to young ones. And here the zygote develop into young ones inside the body of female. Now, the characteristics of the sexual reproduction. It is generally biparental. It involves formation of male and female gametes both. Mostly there is fusion of male and female gametes. Cell division or meiotic during gamete formation and mitotic during development of zygote into an offspring. The offsprings are not genetically identical to the parents. Multiplication is slower than in asexual reproduction, and it may not result in an increase in the number of individuals. Now, the significance of sexual reproduction. It results in the multiplication and perpetuations of the species, and it contributes to the evolution of the species by introducing variations in a population much more rapidly than asexual reproduction. So, this is the end of the reproduction and modes in case of the plants and animals. This is only the introduction of reproduction. From the next video, we will know about each portion in detail. So, the next video will be on the sexual reproduction in flowering plants. Now, if you understand this video, like and share it and subscribe to my channel Anupma Biology Classes. If you have any problem, any question, any queries, you can ask in the comment section below. Thank you for the watching.